Good morning, students. Am I audible?
Yes, ma'am, you're audible.
Students join. Uh, to start with, let's just quickly revise yesterday's lecture. Yesterday, we discussed about the classification of viruses, and there are the two systems of which are being followed in virus classifications that we have discussed. So, yes, Kshitij, can you tell me what are those two systems of classification that are used for virus classification? Yes, Kshitij, please switch on your camera and screen. Yeah, am I audible, Kshitij? Okay, and Devanshi, can you answer this question? Devanshi, is she there? No, she's not there. Namrita? Tanvi?
Yes, ma'am. Can you repeat your question?
What are the two systems of
Ma'am, your voice is cracked.
which have been used right now?
Ma'am, actually, your voice is also quite faint. I don't know if it's just for me, but
No, maybe I would reduce the volume yesterday because of yesterday. I'll increase it. Just let me set down. Yeah, is it fine now?
Yes, ma'am.
Okay. So my question is, Tanvi, that what are the two systems of classification that we discussed yesterday?
Ma'am, Baltimore classification,
Yes, Baltimore classification, and another was the
ICTV.
ICTV classification. Yes. Okay. Yes, Ayushi, tell me what is Baltimore classification about?
Ma'am, the
Yes,
Baltimore
Ayushi.
classification is based on the genome of the virus and the mode of
Yes.
replication and transcription.
Yes. Okay. Fine. So the next question is for let me see the students. Sam, can you tell me into how many classes the Baltimore has divided viruses?
Ma'am, Baltimore divided into 7
Yes,
classes. Uh, those 7 classes were
divided.
double-stranded DNA genome, single-stranded DNA genome, double-stranded RNA genome, and single-stranded RNA genome plus sense, and single-stranded RNA genome minus sense. Then single-stranded RNA with that replicated with DNA intermediate, and double-stranded DNA that replicated with RNA intermediate.
Okay, fine. Yes, Manaswini, can you tell me something about the ICTV classification for plant viruses?
Yes, ma'am. Am I audible?
Yeah, you're audible.
Okay, so basically the ICTV classification has, so it actually is mostly basic properties. So for example, it included, the symmetry of the virus that, the symmetry and shape of the virus. So that was based on the structure of the capsid and the envelope as well. So for example, it could be, based on just morphology, it could be either rod shaped or spherical shaped. And then it could also be classified based on whether it had complex symmetry or helical symmetry. It could be classified based on what it infected. For example, let's say a virus infected skin cells, then it was in one particular category, and if it infected neural cells, then it was a different category. And based for if we're talking about plants in general, then it could also be included based on what kind of plant it's infecting. So for example, let's say we talk about Gemini viruses, then we know that there are these DNA viruses which infect mainly plants, and different viruses will infect different kinds of plants, and also a little bit about the infection that they cause and the symptoms and so on.
Yes. Okay. Good, Manas. And you see, the Baltimore classification, yes, it is based on the genome of the viruses, single-stranded RNA, double-stranded RNA, negatively translate, positive sense, negative sense, right? So if we talk about the plant classification in particular, ICTV that is given, it is again based on many factors, like they take into consideration the genome again, whether they are DNA or RNA. Then in further in RNA, whether they are positive sense or they are negative sense. In DNA also, like whether they are single-stranded DNA, right? So different consideration by classifying the different viruses which belong to the different groups. See, at the onset, yeah, that is very important when we talk about anything. Like when we talk about human, when we talk about plants, when we talk about animals, or when we talk about pathogens, classification is required. So whenever a new pathogen is identified, now where to place that pathogen in the classification, how to that pathogen. So normal classification plays a very important role here. So we know by of a particular virus by looking into its genome, by looking it into its virion size and particle, what kind of a particle it is showing, we can place it under a certain kind of a category or a certain family or a certain group. So this is required. So today's important, it is also a very important topic that we are going to discuss today. It's about virus transmission. So first we'll study plant virus transmission, and in the next lecture we'll study human virus transmission. So there are very different ways, like human viruses, we all know they are transmitted by maybe the aerosols, maybe sneezing through direct contact, or skin, through skin if there are cuts in the skin, right? This is how the viruses we get contaminated or we get infected. So in the plant, it is more complicated in the plant. When we talk about plants, there are a number of ways by which viruses are transmitted. There are mechanically transmitted, then they are transmitted by the vector. And in the vector also, when they are transmitted, there are different ways. They are like persistent viruses, non-persistent or semi-persistent. So I'm going to discuss in detail about these terms, that how different insect vectors also transmit viruses in the plant, right? So they are being transmitted by the aphids, beetles, thrips, nematodes, fungi. So all these are the vectors of the plant viruses, right? So transmission, definitely see, transmission is important for viruses to survive and to maintain their kind, right? This is how they maintain themselves and how they multiply themselves. So it is definitely a fundamental property. Plant viruses, we know these are the obligate parasites. So definitely for them to spread from one host to the another host, they need some kind of a method or some kind of a way through which they can introduce themselves to the other plant, right? Definitely for their survival, it is very important. So why we need to study about the plant viruses? Because it recognizes a virus as a cause of the disease. If it is transmitted from infected to healthy plant, then it helps us to tell that how viruses spread in the field. So if we know that this particular virus, for example, Begmo viruses are the, is my voice okay? It's not going like last time. Is it fine? Yes, ma'am.
Okay, fine. So see, why we need to study this? Because if we know that how viruses are spread, then only we can take some steps for the management of that virus. So for the point of view of a controlling a virus or management of the virus, it is also important to know that how this virus is being spread. Like I was telling you example of Begmo viruses, plant viruses, and they are transmitted by the white fly, Bemisia teresi. So if we can somehow control the or manage the vector by using some kind of a insecticide or any other biological control, we'll be able to manage the virus also. So that's why it is important to know the vector of a particular virus, that how it is transmitted.
then we need to study biological relationship of interaction between virus and its vector, how it is being transmitted, what for what or how long it is being there in the vector, whether it is replicating in the vector or it is just there for some time and then it is coming out. So all those properties we need to study. And the mechanical transmission is again, see, uh, bacteria, if I want to culture a bacteria, I'll take media, nutrient agar media, I'll put directly that starting material, maybe inoculate by using streaking or any other spreading method, right? That is how I can maintain that. But in viruses, as these are obligate parasites, we cannot do that. So we need to mechanically transmit the viruses to a live host plant, right? If from a certain plant I have got the culture of that virus, that this virus is present in this plant. Now I want to study further. So for the maintaining it for a longer time, I need to transmit this to the another plant, right? So in mechanical transmission can be done very easily by using simple techniques. But some cases since vectors are playing a very important role, in some cases mechanical transmission is not possible. So we need to go for insect transmission, and there are certain methods for that also. And to study viruses also, we need, it is important that we should know how they are being transmitted, right? So if we broadly classify them, they are being classified into two categories, which are, one is the horizontal transmission and another is the vertical transmission. So horizontal transmission, it takes place by vectors usually, or maybe by humans while using different equipments or pruning shears and shears tools in the lab field, or other direct contamination. So by those methods, it can be done. So that kind of a transmission is known as a horizontal transmission. Vertical transmission is basically when a new plant, it is receiving its from its parent plant, right? So either through asexual reproduction, cuttings, right? We do a lot of cuttings in the trees and we grow plant by vegetative propagation by cutting plants, right? Or by sexual reproduction by infected seeds. So vertical transmission is basically when a plant gets it from a parent plant, then parent plant is already infected and it is producing seed, and those seeds are infected by viruses and we are using those seeds to grow in the field. So it is getting transmitted. Seeds are also infected, right? So if we talk about methods of transmission, non-insect transmission are basically, like I told you, the mechanical transmission. Transmission, proper method that how we do that we'll do in a practical. So here I will not discuss the method that how we do it. I'll discuss in the practical class, because in the practical class also we have a transmission experiment, right? So but it's very easy if we just need in a normal or in a simple way. If I tell you, it's like you prepare sap out of the infected plant material, you crush it in some kind of a buffer, extraction buffers are there. Then in the plant leaves, I want to transmit, usually are host plants such as Nicotiana tabacum or Nicotiana bantamana or other any normal growing plant which can grow in the field, in the glass house or a polyhouse condition. So I'll just make certain abrasions using carborundum powder on the leaves. I'll select leaves where I want to, and I'll just simply rub that sap that I have crushed that has a virus inoculum on that leaf. And we watch that leaf for, from starting from 3 to 5 days. We know the incubation period of the viruses. So we'll start observing the symptoms. And once the symptoms appear, we'll test it by ELISA or a PCR whether the virus is there. So this is the simplest method for, or we call a mechanical transmission, right? Then insect transmission also takes place in certain viruses, being golden mosaic virus or potato virus Y. So other are graft transmission, vegetative and graft. So what is grafting? Grafting is a technique which is being used. For example, rose is usually grown by grafting, right? There's a the root is another part, and the scion above plant is another part. So grafting is usually done. Why we need to do grafting? Because some species, their root stock is not very good. So we use the rootstock from a wild species, and on that we do graft with some cultivated species. So mostly आपके stone fruits and pome fruits, they are grafted. Their rootstock is different and the main plant is different. So this is the main reason that why we do grafting. So what will happen? What will happen? if a rootstock is virus infected, or the main plant that we are using is virus infected, virus is going to transmit in that case also. So how? By grafting also viruses can be transmitted. Then nematodes, through nematodes definitely viruses can be transmitted. Examples are fan leaf virus. We'll study in detail in the further slides. So mechanical transmission, as I told you, they come when plant come in contact with other plants and leaves, they rub together. Mechanical transmission can take place in the field also. Plants are very close to each other. There is a proper rubbing of the plant. Abrasion will take place and virus can be transmitted. Or it can be done by humans, by knowingly or unknowingly. Mechanical, this I have told you, right? I'll discuss this in detail in the practical, that how to do this, right? Methods also I'll discuss in the practical class. Then we have an insect transmission. No, we know various vectors in plant viruses. They are maximum, they are transmitted by the vectors. They are insects are the most important group. More than around 400 species have been reported to transmit plant viruses, and 94% of these belong to phylum Arthropoda, and 90% are insects. They all are insect. 70% of insects, they belong to Homoptera, in which Aphidae is the aphids. Basically, through this family, aphids belong, and they are the most important group. Others are your plant hoppers, your beetles, whiteflies. Then we have a fungi, we have a nematode, right? So, in general, what happens that viruses transmitted by one group of vectors are not transmitted by the other group. They are specific, right? Or specific, or we can say they are virus specific. That particular virus only they will transmit, except there is a tobacco ring spot virus which can be transmitted by nematodes and by thrips also, and by spider mites also. So different groups. This is the exception. Then these are the different terminologies that we use while we talk about the virus transmission, when we discuss about the persistent, semi-persistent, and other modes of a transmission. So acquisition access period. So what is acquisition access period? It is basically the time for which initially virus free, that means the vector is a virus free and it is allowed to access a virus source if it feed on that particular plant or not. Now it is that particular time. It is the time taken by the vector whether it is going to feed that plant or not, right? Then acquisition feeding period, as the feeding is there, right? So this means the time period which is important for successful acquisition of the virus by its vector, and after then only it will become viruliferous. Viruliferous means it has acquired the virus now. Inoculation access period, the time for which virus carrying the vector is allowed access a virus-free plant and it could feed on it. So there's a virus-free plant and it is feeding on that particular plant. That is the inoculation access period. Inoculation feeding period, the time period for which a virus carrying vectors appears to be feeding on the virus-free plant to transmit it. So these terms, they look very similar, but they are different, right? Different, one terminology is different in each definition. Transmission threshold or inoculation threshold or inoculation access threshold. So that means the minimum initial time period that a particular vector need to acquire a virus and then inoculate it to the virus-free plant. So there is a minimum time period. For example, if the minimum time period for a particular virus vector to acquire virus is 10 minutes, but it is feeding on that plant only for 5 minutes. That means that virus is not virulent. That vector has not, get the access to that particular virus. That vector will not be able to transmit because the minimum time period, it is not feeding the plant for that minimum time period, right? Infective capacity or retention period of the vector, the time period for which a vector carries and retains and transmits the virus to the host plant and it remains virulent. Incubation period or a latent period. This is the time period from the start of the acquisition feeding period until the vector can infect the healthy plant with the virus. So these are the different terms that we are using further also. Now, virus vector relationships or types of transmission, basically the different types of the transmission that based on the virus retention time by the vector. They are non-persistent virus, they are persistent virus, or they are semi-persistent virus. Now, virus-vector relationship is also based on the site of the retention of the virus in the vector. Whether they are stylet borne, that means they will just remain at the stylet of that insect. They will not go beyond that. Whether they are circulative. Circulative means the virus, they circulates up to the food canal, across the midgut and hindgut, and then they reach the hemosyl. Then there are propagative viruses. Propagative viruses, they are able to replicate both in plant and in the insect also. Then transmission, if there is any transporial transmission also, right? So if we talk about the first group of viruses, which are called non-persistent viruses. So as the name suggests, they are not there in the vector for a longer time, right? Non-persistent. So such viruses, they are acquired by vector during probing. When probing means when they are feeding on the plant, and a host parent gamma, they go to the epidermal cell, and probing takes as little as 5 seconds, only for 5 seconds. So vector becomes infected immediately after the feeding. Virus lost by the vector during molting. So molting in a process, arthropods या insects में molting is a process. It is basically the shedding of the exoskeleton. So during that process, the virus is lost. उससे पहले अगर उसने healthy plant में feed किया and transmit किया तो it is fine. Otherwise, it is lost. There is no latent period. Such viruses are mechanically transmissible. Acquisition fasting increases acquisition of virus transmission. So if in the lab we are doing this kind of insect transmission, and we keep these vectors in a fasting period, maybe 1 day or maybe 5 to 6 hours, and after that we allow them to feed on a healthy plant, that it increases the virus transmission. This type of viruses are basically cucumber mosaic virus, bean common mosaic virus, potato virus 5, and papaya ring spot virus. So there are a number of viruses which come under this category. Then we have a semi-persistent viruses. So as the name says, semi-persistent, right? So they can persist in its vector for 10 to 100 hours. It is very elongated time. It is acquired from phloem region with a long feeding. They do not circulate and multiply in its vector. And again, in this, infectivity is lost during the molting. They accumulate in a particular sites. They have a high vector specificity. For example, citrus disease virus, cauliflower mosaic virus, these come under these kind of a category. Then we have persistent viruses. They persist in the vector for more than 100 hours, and in some cases for the whole life of a vector. They are there with the vector always. They viruses now. In this case, viruses, they multiply and circulate in the vector body. Molting has no effect on the virus after virus uptake. It goes to the elementary plant, gut wall, it circulates in the body fluid. Example is bean yellow dwarf virus. So persistent viruses are the most dangerous ones because they are there in the vectors for the longer time throughout their lifetime. They multiply also there, and molting also does not have any effect on them. Then persistence transmission, again, this type of a virus transmission in which virus is acquired and transmitted by vector after a long feeding time. So definitely when the feeding time is long, they are retained in the viruses, vectors for a longer time. Then circulative transmission, again, it is characterized by long period of acquisition of the virus by a vector, and there is a latent period of several hours, right? So before the vector is able to transmit, so it need a gap. It is not like it is feeding on the plant. Now it needs a latent period of several hours before it is able to transmit the virus to the plant. So if it is after immediately acquiring the virus, it is going to sit and feed on the some plant. So it will not going to be transmitting the virus. It is just feeding. Virus is not, because for virus transmission, the latent period of several hours is required. The virus circulates in the body of the vector. Aphid as a vector, as I told you, number of vectors are there. More than 370 species, transmitting more than 300 viruses. Mysis persicae is just one example. There are a number of species of Mysis persicae which is transmitting along more than 100 viruses. These are features of aphid transmitted viruses like how they are transmitted, they are mechanical, whether they are persistent, semi-persistent, or persistent. What are the common symptoms which are being produced? Mosaic, yellowing, leaf rolling. What are the tissues of virus? Like some viruses, they are going for epidermis, and then the mesophyll, and the mesophyll and phloem. What is acquisition time? So these are the certain characteristics of the vectors. Now there's another term which is called as a helper viruses. Now what are helper viruses? These are the viruses which are transmitted by aphid vectors, but under certain conditions. Aphid can only transmit the virus if the source of the plant, that host plant, is infected by a second virus. Now, this second virus is called a helper virus. So it's a dependent virus, and the second virus is reflected as a helper virus. So there are certain plants which have two viruses. One is dependent, and one is the helper virus. For example, this is the potato Acuba mosaic virus. It has a PVA, potato virus A or Y, as a helper virus and transmitted by the Mysis persicus, and it is a non-persistent mode of a transmission. Then carrot mottle has a carrot red leaf as a helper virus transmitted by, again, Aegypodi species in a semi-persistent mode. So these are just the example of a helper virus. Then whitefly. Whitefly can transmit the ghost disease. It is whitefly, let me tell you, the whitefly is the most abundant vector which is found in the field, and this is usually found in the warmer regions. And we'll find more whitefly in the glasshouse or polyhouse cultivation because their temperature is little higher. So I don't know if you guys have happened to be in any field anywhere, but if you just see the underside of a leaves in the plants which are growing in a glass house or even the field conditions also, you will find white colored small, very small flies sitting in a large numbers there feeding on the plants. They're numerous. So they are very acting as a very important vector for transmitting any virus. And believe me, it is very difficult to control or manage whitefly borne viruses because to manage a whitefly, it itself a difficult task. There are thousands or lakhs in numbers in the field. We cannot do anything. The whole plant is sometimes covered by these whiteflies if they find a suitable temperature and environment, right? So Bemisia typica is the example here. They are semi-persistent to persistent, and the females are more efficient in transmission. Within period of few hours, they can feed in the phloem of the plant and virus not transmitted by the sap. So whitefly borne viruses, for example, Begmo viruses, we cannot transmit it by mechanical inoculation. We need to rear whiteflies. We need to feed them first on the infected plant, and then we need to feed them on the healthy plant. This is how insect transmission is done. Tobacco leaf curl virus, moonbeam yellow mosaic virus, all big viruses are transmitted by whiteflies. Then we have leaf hoppers, more than 30 viruses, not many, but yes, around 30, 35 viruses, semi and persistent transmission, propagative viruses, leaf roll and yellowing symptoms, they go to the phloem cells. Leaf hoppers, for example, rice stalk virus, right? It is transmitted by Nephotratis. Then rice tungro virus, again, Impicite cepus virus vector. Transboreal transmission, again, in the rice stalk virus. So transboreal transmission, it occurs in arthropod vectors where they transmit pathogen from parent arthropod to the offspring arthropod. So that kind of a transmission is called a transboreal transmission. Then we have thrips viruses in the genus Roscoviruses.
Excuse me.
Yes, Adesh.
Ma'am, are whiteflies and white mealworms different?
Yes, yes, they are different. White flies are different. Worms are worms, and white flies are flies. Insecta में आएंगे.
Ma'am, I
Worms तो
always
insecta में
thought
नहीं
them to
आते.
be their larva.
Pardon?
Ma'am, I always thought them to be their larva.
No, 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 no. White flies are different. White flies are different.
Okay, ma'am.
So white flies, see, white flies are basically, as I told you, white flies like warmer temperature. They are found in the tropical areas. They have more than 30 or 40 biotypes, means different white flies which are available and which can all transmit the viruses. And the now, if I'm not wrong, maybe it's long 10 years back or somewhere, that white flies or Begoviruses were found. Earlier Begoviruses, they were found only in the tropical areas like India also, the warmer areas, but maybe in 2008 or 9 from my lab, only my colleague, he identified the Begoviruses in the temperate regions like in Himachal. Himachal has a temperate zone. Yes, in some part of the Himachal is comes under tropical zone also. They have a warmer climate, but still then it was found in the temperate zone at that time, and that was the first. This was the Palampur, if I'm not tomato leaf curl Palampur virus, right? So that was the onset. I mean, that was the beginning of the, white fly. They are changing their environment, moving to the newer environment and adapting to the newer environment. So when these kind of a transitions happen, or when these kind of a changes happens in the vectors especially, it is harmful for the crops, because earlier though at that those places those flies were not present, so those crops were not infected by the Begoviruses. But with the vectors changing themselves to the and adapting themselves to the lower temperatures, that means those plants or host plants, they are becoming access to the white flies and definitely the DNA viruses or viruses. So it is whenever any, see, when we talk about changes in the viruses or mutations in the viruses, right? We always say that it is not very good for the humans also. Similarly, vectors, they are the main source of the virus transmission in the plant. So if they are adapting themselves to the different temperatures or different climate, and they are going to the places where they were not found earlier, so it is a negative sign for the growers that they'll come with the baggage of viruses or pathogens which they can transmit. So these things have a negative impact on the for the growers especially, right? So thrips also transmit the viruses in a different groups, rhinoviruses and wheat streak mosaic viruses. Then we have beetle transmission, acquisition feeding up to 24 hours. So that means it needs to feed for at least 24 hours on that plant, and then it will transmit by persistent transmission. Cowpea mosaic virus or turnip yellow mosaic virus are examples. Then nematode transmission. See, nematode transmitted viruses are not much, they are still there, but they are important, right? Nematodes, you understand, nematodes are the pathogens basically. We call them a pathogen. They are found in the soil, right? They are found usually the roots, or they are found near the roots of the plant in the soil. So they affect the plant also. Nematode, sometimes they go to the upper part of the plant also. That's why we say that we should wash cabbage and cauliflower, those kind of a vegetable very carefully, because they could be the nematode infected. Again, they act as a vector of the virus also. So it was first identified in 1958 that they act as a vector, and how they are transmitted, soil-borne plant virus disease basically. So usually two genera, Nepovirus and Tobravirus, they have nematode vectors, and Tobraviruses, tobacco dettel virus, they have nematode vectors. Again, I'm not going to discuss in detail about what are Tobraviruses. We are talking about the transmission here, right? Then Nepoviruses are nematode transmitted polyhedral viruses. Again, the only known nematode virus vector are in the genera Zipinema and Longidorus. So only two genera of the nematode, they transmit these nepoviruses basically. Then vector specificity, they have a vector specificity also. In 11 species of Zipinema, they transmit 13 nepoviruses. 11 species of Longidorus, they transmit 10 nepoviruses. 14 species, they transmit again 2 strains of a tobravirus. So it shows their vector specificity also, that how they are transmitting different kind of viruses. Okay, transmission by fungi, it is not to that extent, but yes, there are some fungus such as Olpidium brassicae, which is parasite virus. And there's another example is Polymyxa graminis, which also transmit Brimovirus, Nicolovirus, or a Plomovirus. They are not very important plant viruses, but yes, it is transmitted. Then again, the question is that the specificity of transmission by vector. How and why? So why the vector of most viruses are confined to one taxonomic group? Why the tobacco mosaic virus is not transmitted by homoptera, and why different strains of a virus may differ in vector transmissibility. So we cannot have a proper answer to this, but there are certain factors which are involved here which might answer this. That transmission is there are a lot of, see, today we have just discussed the, overview of a transmission. Other, if we want to discuss that how transmission in a viruses, how viruses are being transmitted in a plant, how it goes through the plasmodesmata and the phloem and the moves. So that is the another part, right? So it is a very complex process. It is not as easy as it seems that persistent, semi-persistent, non-persistent. No, there's a lot of things. There are a lot of pathways which are involved here. So acquisition of a virus by vector, then how survival of infectivity during association of a virus and a vector. This is also one factor. Then inoculation of virus particle to the plant, which is followed by the initiation of infection. So if any of these processes does not take place properly, it will result in no transmission. Basically, the virus will not be transmitted in a proper way. Then another possibilities are that non-vector may fail to acquire virus because it does not feed on the appropriate tissue. That could be the reason. Then plant species is different. It is not the host of particular vector. Then that are the host of the virus. I mean, non-vector host of it is not a vector. That could be taken up by non-vector, but then they'll not be transmitted because it is not a vector of that particular virus. So there could be many reasons. The host could be the reason, the virus could be the reason, the vector could be the reason for this, right? For example, in case of fungi and nematode vectors, there may be specificity for the attachment of the virus, and which again depend on the protein coat and other property of the virus particle. There may be certain transmission factors which are absent in non-vector. Virus core protein seems to play a very important role in vector specificity, especially with viruses that do not multiply in their vector. So core protein here plays a very important role. Then aphid saliva may also influence the transmission efficiency and specificity during probe, means during the time when they are feeding. The polyphenol oxidase activity of the sheep saliva may affect the virus. Then at the same time, physiology of the host cell may be affected by salivary phenols and phenolics and carbohydrate hydrolyzing enzymes. So number of factors are there that takes place and due to which the virus transmission can be affected. So we need to understand the biology of vector transmission, what is the, how it is being transmitted. So those things can reveal certain answers that how and what are the different pathways through which this transmission is taking place. Are there any specific receptors present which are important for the transmission of the virus? How a host specificity is there, how a particular virus is vector is identifying that it is particular host, or how it is identifying this is a particular virus that I need to transmit and this virus I can't transmit. So there are many factors which are involved there. Then there is another kind of a transmission which is known as a bimodal transmission. So while aphid transmit non-circulative viruses, either non-persistent or semi-persistent way, there are viruses which use both the ways for the transmission, right? So this typical mode of transmission was rapid as a bimodal, and it is being identified in cauliflower mosaic virus by Myges persicae and this is Brevicornia brassicae, two different species. Then seed transmission, around 1/7 of the viruses are transmitted to seeds, and they help in dispersal across a longer area. It is very economically important. They help in virus survival. So if particular virus, they are going to the seeds are from the plant that we are saving maybe for the next season also. Like some of the lot is, maximum lot is being used as a vegetable or whatever way we need. And the some lot from that we keep it for the next season. So if it is virus infected, then definitely that seed is going to, when we are going to sow that seed in the field, the plant will be definitely a virus infected in the beginning only, right? Then the seed transmission can occur again in maybe two ways: externally seed borne due to external contamination of the seed by particles, internally seed borne, internally it is inside the seed, and due to the infection of the living tissues of the embryo, then virus may be found in different part of the seed, but generally in the embryonic tissue. So they become infected by directly from the mother plant by the pollens. So developing embryo can infect it before fertilization by infection of the gametes or by direct invasion of the embryo after fertilization. So virus, it moves through the testa of the seed after fertilization and reach the micropyle region of the embryo, infection to occur. So this micropyle region is in close contact with the embryonic substances that helps in the nutrient flow to the embryo. So this is how it is moving to the embryo. And if it is in embryo, so definitely the seed that will be going to form, it will be there also. And distribution of virus within the seed, transmission due to surface contamination of seed or viruses in the seed coat is rare, and it is found in viruses like tobacco mosaic virus. Most of the transmission occur when embryo become infected. So virus is distributed random and can be found in embryo, endosperm, or a seed coat in a different part of the seed. Embryo being common mosaic virus, endosperm Barley stripe mosaic virus, again seed coat, tobacco mosaic virus, tomato spotted wilt virus. So a different part of the seed also, it is a different kind of a viruses can accumulate. Factors affecting seed transmission, it varies with the different viruses strains, host plant and variety. For example, bean common mosaic virus, 90% in different varieties. Being stripe mosaic virus up to 75% only. Then time of the infection is also very important. Location of the seed on the plant, age of the seed, temperature, or if there's any host resistance present. So that also affect the transmission of the virus. See, host resistance is a very important factor. And when we talk about virus-free plants, we use this phenomena of the host resistance plant, and we use in this case, we use the plants which are resistant to the viruses, and then we can, breed these plants with the known variety which is infected by the viruses, and we might get a variety which is resistant to a particular pathogen. So classical way of introducing resistance to a non-resistant plant for a particular virus. Or if we talk about the modern technique, then transgenic is the way. So we identify in a resistant plant, we identify the genes which are responsible for the resistance, which are giving resistance to the particular plant. So we can use those genes, we can transmit, we can just put those genes in the new plant, or the plant which we want to virus resistant, and plant which we will develop will be resistant against those viruses. Usually the common way of developing virus resistant plant is a pathogen derived resistance that we use. So that we'll see if we can discuss, or it's a part of, I don't think so that's part of your course, but in the management we can discuss pathogen derived resistance and cross protection techniques which are being used in case of a plant viruses, right? So this is all from my side in today's lecture. Transmission is not very complicated, but yes, you need to learn the terminologies. You need to know the particular terms that we are using, like persistent, semi-persistent, circulative, non-persistent, propagative. So acquisition period, all these terms. So you should try to just go through this lecture once again and try to see if you still feel that it is tough and you're not understanding, we can take it maybe once more, but I don't think it's that difficult. It's just the overview of transmission. I'm not telling you the molecular biology of transmission that is not in your syllabus basically, that we teach in the master's level, but just an overview that how viruses are transmitted, what are the different vectors of viruses which transmit different viruses, how they do it, in what way, what are the major means like persistent, semi-persistent, or non-persistent ways of a transmission. So just a little overview of the virus transmission. So if there's anything you need to ask, please you can ask the question. It's open for discussion. When we talked about the factors which affect the seed transmission, we talk about the age of the seed also, and the temperatures also, because these are the different factors. See, when we talk about the pathogens infecting plants, uh, there are particular time in which the pathogens are more active, like the younger plants. Because why? Because the younger plants are more prone to the infection. In younger plants, see, most of the aged plant, they have developed some kind of a phenolics or some other metabolites which are not liked by the viruses basically. So younger plants, they don't have all these things with them. So mostly pathogens, whether it's a fungal or bacterial pathogens, they love to infect the plants when they are very young. So that is why if that stage is escaped, like the younger plant, maybe a 10, 20 day, 1 week, only 1 month plant, at that stage, it is escaped from the infection of any pathogen, whether it's virus, fungus. So the losses caused, even if at the later stages the pathogen effect, the losses caused are not that much as if it is there already when the plants are young. Because it happens sometimes that at the later stages, the plants, they they somehow they prevent the infection to occur, or there are certain kind of chemicals or compounds in the plants which affect the transmission of the viruses by the vectors or viruses also. At that particular time, they don't go and infect, right? Certain chemicals are being released, there are certain metabolites, many factors are there, right? It happens there, right? So that's all for today's lecture. And I've given you the assignment, the topics also. And see, marks are basically, I will give in, yes, it's a teamwork. I'll see there will be marks like for overall presentation, then the individual marks that how individually you are presenting, how individually you are speaking on that topic. So marks are for that also, right? So prepare yourself in this Thursday. We'll make sure your camera is working, make sure your mic is working, because you need to switch on your camera also during that time, and make sure that your internet connection is also fine, right? So please switch on your cameras for attendance. Adish Mishra,
Present, ma'am.
Athoiba,
Present, ma'am.
Ayushi,
Present, ma'am.
Sam,
Present, ma'am.
देवांशी.
प्रेजेंट मैम।
आप लेट ज्वाइन किए थे देवांशी, मैंने आपका नाम लिया था, आप थे नहीं सुबह।
मैम मैं क्या कहते हैं, मेरा टीम्स हैंग कर रहा था तो मैंने फिर लैपटॉप से ज्वाइन करा था।
इशिता अरोड़ा,
प्रेजेंट मैम।
जानवी सिंह,
Present, ma'am.
क्षितिज सिंह,
प्रेजेंट मैम।
क्षितिज, मैंने सुबह भी आपका नाम नहीं लिया था, आप सुनते नहीं हो बिल्कुल भी। मानसवनी श्रीधर,
Present.
मानसवनी, are you there?
Yes, ma'am, I'm
नम्रता पाल,
present.
Present, ma'am.
नम्रता, नेहल जोशी,
Present, ma'am. I said. Present, ma'am.
रूपल, रूपल, साहिबा, संजना, can you hear me? संजना, okay. संजना के बाद शक्ति,
Present, ma'am.
शिवानी मिश्रा, तन्वी,
Present, ma'am.
त्रिदिव,
Present, ma'am.
विवेक,
Present, ma'am.
युक्ता,
Present, ma'am.
युवराज,
प्रेजेंट मैम।
okay, fine. So we have a class on, then tomorrow we have a class, right? Okay, I'll see you tomorrow then. Thank you so much. Loving.
एक्सक्यूज मी मैम।
Excuse me, ma'am.
Yes.
मैम
Ma'am, in
Can you please let
Yes.
me know that how many groups are going to present on Thursday? Because from our group, Athoiba has had an exam on Thursday.
Excuse me, ma'am.
Ma'am, you are on mute.
Okay, fine. Go on.
Ma'am, I was just asking that how many groups are going to present on Thursday because
Your group is which group? Group number?
I guess second or third.
I'm group 2.
I guess.
युवराज ग्रुप 2 में कैसे होता था? आपका तो रोल नंबर बहुत एंड में नहीं होगा?
No, ma'am. I have roll
It's
number.
according enrollment number.
Okay, enrollment number के accordingly. It's fine. You can present next day. Maybe on Thursday we'll have maximum 2 groups, 1 and 2. Okay. So let's keep 1 and 2 for Thursday. But we have 2 classes Thursday, right?
Yes, ma'am.
2 classes में at least 3 groups we can present. So we can keep 1 and 2 and then another group जिसका exam नहीं है. Your group we can take to next day.
So our group is 2. And Athoiba is
Athoiba
having
group.
exam. Yes, ma'am. And
Yeah, ma'am.
Athoiba is having exam on 12th.
So, yeah, okay, fine. Then 1, 3, and 4 can present on Thursday, and one group will present on Friday or next week. I'll let you know that time, right?
Excuse me, ma'am.
Yes, Shivani Mishra.
How many slides?
See, you have to speak for at least 15 to 20 minutes and then 5-minute discussion. So accordingly, it depends on you. If one slide you can speak for 3 minutes or 2 minutes or more than that, so calculate according to that.
Okay, ma'am.
Right? Okay. Okay, students. Thank you so much.
Thank you.
Thank you, ma'am.